Okay, thanks for the nice introduction. I think it's really happy to hear the previous talk has already talked about the long read RNA sequencing, so it will be much easier for my talk, hopefully. So hello everyone, I'm Silvia. I'm an assistant professor from UPs. So today we're more talking about after the assemble or like alignment, we can do to play with the long read RNA-seq data. So as the Nature method has nominated, this is the method of year of 2022. So it has great potential for the long read area. So why we do long read, especially long read RNA sequencing? So as we all know, short read has the limitation of the read lengths. So they are not able to cover the full transcript at the RNA sequencing level. So I'm drawing this figure making it so long. So because they have so many strengths. So first of all, they can detect alternative splicings, as I shown in this picture, where if like a multiple isoforms are from the same gene, we can identify those specific isoforms. The other way around is we can detect novel ones. As shown here, if the reference is exon 1 to 3, we can detect the ones like a skipping exon, additional exon, or with some a truncation or extension. So the other way will be like the fusion transcripts, which is defined like originally the two genes are two separate genes, but somehow they fuse all together at the RNA level. But specifically, this is RNA, so we can still define, or we can quote fusion isoform so that we can define the fusion at a more isoform specific level rather than just the gene level. So that's all the advantages of the long read sequencing, and this is our tool aim to address. So we have a tool called IFD long. So we name it as a isoform and fusion detector for long read RNA-seq data. So this is our pipeline. So basically, we were taking all the long read, so it's platform free. So as long as it's in FQ format, like PikeBio, nanopore, or even like some short read, like a linked long reads, we can take in those reads, and then we can take in the reference genome, the annotation file, and then our pipeline, basically, we're doing alignment. We are not a new aligner, but we incorporate some known like a long read aligner, such as Minimap2 or StarLong. And also after that, we can do the annotation. So it can be as some known isoform or some novel isoform, or the other way will be some fusion transcripts. And then the next most important thing I want to say is that we will do some isoform or fusion quantification so that we will quantify the expression intensity of the isoform or the fusion transcript. And definitely our output will be, we will have a long read-based annotation profile, and also the expression metrics as well. So I will maybe not go over all the details, but basically long reads, they will have some truncated long reads as the red one shows, which they may potentially align to more than one isoforms, as the red arrow just show, maybe isoform 1 and 2, maybe their potential targets. While we also have some complete long reads, the purple one, which can usually uniquely align to a specific isoform. So our purpose is we want to develop an algorithm we call the exploitation maximization, the EM algorithm. So we want to quantify the abundance of each isoform, even for the same gene or across the whole transcriptomes. So I will skip a lot of details, but basically we're using some survival models because we assume the long reads is kind of like a truncation events. And then by the iteration, we will estimate the abundance ultimately. So here are some performance. So for isoform quantification, we do some large scale, like in silico simulation data. For example, we do the whole transcriptome expression. Like we are starting from a real human data, and then we simulate their isoform expression and use that as a reference so we can simulate some isoform expression, the FASTQ files, and then we're applying our tool, the IFD long, our pipeline, as well as 3 other popular tools to do the isoform quantification. So as shown in this figure, we show all the pairwise comparison because this is simulation data, so we know the truth. So we can compare, we can check their scatter plot showing their pairwise correlation. We can also check their Spearman's correlation with the truth. And ultimately, our tool, also the LIQA, both shows good way to go. So our simulation, we simulate a lot of different scenarios, like different the sequencing depth, accuracy, and also the mean length of the transcript. So we take into consideration a lot of like factors. So in our second simulation, this is not a large scale, but a very interesting one, because we want to show the power of the long read that if we say this gene, we actually they have 16 isoform, but some quite similar, so we combine. So ultimately we have 12 types of isoform, all sourcing from the same gene. So we want to see the ability of our software that can identify different, this can distinguish different isoforms, even they're from the same gene. So this is the true isoform assignments, while the column is our tool, the annotation. Well, we are expecting everything on the diagonal, ideally, but actually our software showed really good performance because they are quite agreement with the truth. And we also show the percentage and their cosine similarity as a measurement of different tools compared with the truth. So the other powerful part of long read is doing the novel isoform identification. So our tool, we are trying to work that out. So I show this figure of defining all the novel isoforms, and then we define some buffer region to define novel ones. So as you can see in this figure, we are doing a simulation, checking their specificity, sensitivity and specificity for different types of novel isoforms. And it shows that adjusting some buffer regions, we can quite well balance the precision, like the sensitivity and the specificity. We also have some real data applications. So our pipeline can be applicable to human data and mouse data. Here we show an example, like this mouse tissue is from the heart tissue from like 14 days, which is just a transition from their fetal to adult stage. And then our program can successfully identify some key isoforms. We're showing their expression level at their adult stage or at their fetal stage, showing their the heart developmental stage. We can also apply into the single cell data. So not only single cell short read data, but single cell long read sequencing. This application is into like an HCC, like a liver cancer samples with paired tumor and normal cases. So you will see this figure very familiar, usually generated by the short read strategy, but this is like go by the long read isoform expression based, doing the dimension reduction. And we are able, again, doing some pathway analysis. And again, we are able to define different isoforms. So this example is a immune CD47 gene, where we can identify all the 7 isoforms from that gene, and some isoforms showing similar pattern, which all like showing increasing trend in the tumor cells, while the other gene like almost showing pretty low expression level. So that's the additional power of this tool. So in terms of fusion transcript, the third part, we again do the simulation, so we can do the fusion quantification so that we can compare different tools, including the truth and our tool and 2 other like popular fusion tools. So we can compare the Spearman's correlation with the truth. And then this is another simulation, like usually the fusion, there will be some popular mate where like a gene maybe is like paired with multiple genes in the fusion detection. So we're trying to detect if our tool has the power kind of like to detect different mate genes fused with the same gene. So these are some like a distribution of different, the proportion of different mate genes, while our tool in general show overall the best. Definitely we have the real data application. So we have some colon cancer, the HCC liver, some cell line, all go by the bulk or single cell data. So we are able to compare our method, the IFD lung, with the Genine and the JAFO, 2 other popular tools, and in terms of detecting the fusions. And also for fusions, there are 2 segment fusions, and there are also some interesting discovery of 3 segment fusions, where like 3 genes, though they're rare, but they fuse all together. So it all shows that our tool overall can detect these rare events, while some tools maybe fail for all the 3-way fusion detection. So also specifically in our previous publication into the liver cancer, so we're able to detect 3 novel fusions, and we also do like the PCR and the Sanger sequencing validation, and we prove that they're all real discoveries, not all the false positives. So which really shows the great application of our software. Okay, So as a summary, our tool, IFD Long, is an isoform and fusion detector on the long-read RNA sequencing data. And our tool is available at GitHub. So we just have a preprint at the bioRxiv, and we're aiming for peer review submission. So overall, the function will be isoform quantification, novel isoform, fusion transcript, and then we have application to like a bulk samples, application to all the single cell samples where those work has been already published by eLife and the communications biology. Okay, I would like at last to acknowledge all the people who has helped with this project. So people in my lab, we have a PhD candidates, I have a master student, and also like a statistician in my lab. And I also want to thanks for my collaborators who give me a lot of suggestions on the liver, on the heart tissue applications. And also I would appreciate the funding institutes, several like internal pilots grants, and also some like big external or foundation grant support. So I'm always actively looking for more collaborations with the people, making a self-advertisement, and welcome any question. Thanks for attention. Yeah.
Great. So we have a couple minutes for some questions, if there's any from the audience. Again, if there's questions, either talk really loud so the mics can pick you up, or walk down to one of the microphones.
Yeah.
Yeah, I would ask, what is percentage of libraries you think are truncated in your data?
In truncation, yeah.
Truncated like fragmented libraries.
Yeah, that's a good point. I think it's very like platform dependent.
Yeah.
I would say usually like a PacBio has a like a lower truncation rate because its hi-fi rate is just circulating it and sequence again and again. So I think the rate is overall low. I think PacBio is okay, but I think for some short read linked method, maybe the truncation rate is long because they may only have some like middle part linked while some tail part are missing. Yeah.
How about nanopore?
Yeah, nanopore I think is in the middle of PacBio and those short read. Yeah.
Are there any questions online? No. Well, if there's no other questions, yeah, Jane.
Yeah.
Could you comment? So we all face the problem that if we can simulate data and know the right answer, but simulated data doesn't perfectly capture real data.
Yeah.
Or we can apply it to real data, but then can you comment on how that plays out specifically for long read assembly?
Well, yeah.
And what kind of simulations you use and where the limitations are?
Oh, yeah, that's a very good point. Yeah, because as you just say, like simulation data cannot reflect the whole picture. So in our real simulation, we actually find some like the universal human reference and we quantify their expression. So our isoform expression intensity actually simulate their distributions. So we want to try our best to reflect the real data. And in the meanwhile, we simulate different like accuracy level because there are popular platform like PacBio or Nanopore. They all have different like accuracy because long read the most concern is the accuracy. So we actually simulate different accuracy level to reflect the real data. But I have to say, simulation data definitely has their limitations because it's never perfect, and our truncation sometimes is hard to track the real. For example, the truncation where it's missing. So that's all the limitations. So that's why we go by real data. But real data is the real. But on the other hand, like we don't know the real truth of the real distribution. So we're using, for example, short read sequencing, or there's a like say we're using the universal human reference because there's a project has around like I remember like 1,000 isoforms being quantified by the PCR method, where we regard it as a golden standard of the truth. So we compare our long read sequencing and doing the quantification and compare with the PCR method and see how consistent the agreement of those two. Yeah.
So does that mean tools in general are all optimized for human and perform suboptimally on, say, fungi?
Yeah. So currently our
and
tool.
Not just your work, but
yeah.
Is that a problem generally?
Yeah, I would say not like a human, but I think it can be generalizable to maybe mouse data, like our rat. But we are not specifically working for, say, fungi, bacteria. Those maybe not such optimized. But for mouse, rat, human, I think they're kind of like similar strategies. Yeah, yeah. Yeah.
Great.
Okay, yeah,
So it's like Sylvia again.